Today I have two sessions to present for you. One is genetic engineering and another one is gene therapy. Two sessions. The second one is therapy, means what we do in our job as a medical doctor. But this one, genetic engineering, to be honest with you, is absolutely different way. When we are speaking about genetic itself is a science, such as biology. When we say biology, means just science. But when we say biotechnology, is not science, it's technology. So when we say genetic engineering is a technology, it's not only science. When you say genetic is just science, when you say medical genetic or genetic engineering, they are applied science. Look what we have. First we have research, for everything, any kind of science. We have research. The results of research is science, something you discover, and then after that you change this one as a technology. Using this technology, you produce something, and the end of that is what? What? Money. To be honest with you, this way is for money, nothing else. All of us. I don't know about you. It is for me. So I don't say about you. You are very good people. But I go this way. You go to God. You go to, I don't know, somewhere else.
We will also follow you.
I don't know. You go for people, for God, for country, for something. I don't. I go exactly for money. Is it clear? Very clear, I suppose. That's why when we speak in our teaching, our study, you study many subjects. All of the subjects, research is science, technology, product, any kind of product. This is not just only product, maybe it is services, product or services, and then after that is money. The money coming from this way is, to be honest, is a good money. It is the best one, because this is from science. That absolutely it is knowledge-based earning money. Knowledge-based earning money. What I am going to say today, exactly in this lecture, I am speaking about this part. Changing science to technology to product, and product to money. Is it clear? That's why we are speaking about genetic engineering. Okay. What we speak here, definition, history, processing, application, we discuss about this one more, and research and industry. Genetic and very quickly, genetic is part of biology, we know very well. Biotechnology, as I said to you, biology is science, biotechnology is not science, it's technology. Absolutely different attitude, different view, different attitude of us to this subject. Genetic engineering, what's genetic? Genetic is genetic, as we read, we can read books and anything. But when we say genetic engineering or genetic modification, means you do application, you do something and change some genes and some DNA and something for some reasons, such as product. What is biotechnology? As I said to you, what's the difference between biology and biotechnology? As I said, this is science, that one is technology. I explained that one. And now I am trying to explain more about the DNA and hosts. What's host? You are going to produce mobile phone. For example, mobile phone. What do you need? A factory. You need a factory. You cannot produce in your home. You need a factory because you want to produce a lot of mobile phone. What's the difference between this factory and our factory? Our factory is a host organism. Do you understand what I mean? We use bacteria, we use animal, we use plant as a factory. Understand? Understand? Not human. It is not ethical. It is not ethical. But we use animal, we use bacteria, we use different hosts, host organism as a factory. What does this mean? This means we produce whatever we want in the bacteria, or in an animal, which I want to explain for you. How by genetic modification of the organism. Genetic modification of the organism. Which organism? Host organism. So, we produce something in that organism. Do you remember I said? Science, technology, product, and money. I am speaking between these two. Is it clear? Okay. How we manipulate? Anyway, we focus on a gene and put the gene in that organism. You remember last session I said to you DNA, RNA, protein, and DNA, RNA, protein, and function. Do you remember yesterday? Okay. What we do here, the organism has got DNA. If I change the DNA, I can produce my favorite RNA, my favorite protein, and my favorite money. Not function. Function is for somebody who buys the protein. That's the money. Is it clear? Okay. So, the gene, we can put it here. That's the gene. What we say to this organism, we say transgenic organism. Transgenic. Because I transfer a gene into the genome of the organism. We say transgenic organism. Okay. Let's go to history. History is from 1952 and up to now. We have a lot of cloning animals, cloning plants, and so on and so on. I want to give you some example, just example of methods, because we use methods to change the DNA. The easiest method is PCR. You know PCR. After Corona, everybody knows PCR. Even my grandma knows PCR. Before that, nobody knows PCR. But after Corona, everybody knows PCR. Am I right? Yeah. So what is PCR? Polymerase chain reaction. Anyway, I suppose you know PCR. For PCR, we have okay. For PCR, that's the DNA. One DNA. This DNA, we want to amplify just part of genomic DNA. Look at that. This is genomic DNA. This is genomic DNA. We are going to amplify this one. Can you see DNA? No. But I amplify, because when I want to manipulate, I have to see. I amplify this part of the DNA, just this part. How I can? By PCR. How? I design a primer here. I should say here. Primer here. According to these two primer, I amplify this one, like that one. Denature, do you know DNA is double strand? Denature DNA, put primer here, and add DNA polymerase. DNA polymerase make two strands of DNA, doesn't matter. And after that, what happened? I have two, and again after each cycle, two is doubled. Next cycle, two is doubled is 4, 4 doubled is 8, and change to double. Do you know the difference between exponentially and linear? Linear is 1, 2, 3. Exponential is double, double, double. Do you know chess?
Yes.
Everybody? Who is the best? Chess. I saw in your hostel. The chess. Who invented chess? Who? You don't know?
Yes, I know.
Who? Who invented chess? Chess.
Long,
Long.
Which was the question?
I think Chinese.
It was, to be honest, from India, but India was from Persia, long ago. And he came to the king for to invite chess to get prize, and asked the prize, and king go, okay, give the prize to him. He knew mathematics and said, give me just one wheat. You know wheat?
Yeah.
Wheat. Just one wheat. Okay. But put one in each box and make it double in next box. How many boxes we have in the chess?
64.
64. And said, put one here and double to next, double next, double next. And the king said, okay, it's nothing, just wait. Give him the wheat. But didn't know about the exponentially increasing of that. So after said to people, go and give it the prize. And they back to the king and said, do you know how many grains of wheat it would be there to the 64 bucks? is this side, this one. it means for 7 years, whole the world, if they grow the wheat, they cannot provide this one. Understand? That's increasing of exponential. When we are speaking about PCR, is exactly the same. Understand? This is a story. But somebody from this story got the Nobel Prize. It's exactly the same as this story. But the person who found or made PCR got Nobel Prize. Do you know why? Because he knew about this one. Understand? And not only Nobel Prize, also he patented this one. I got many money.
But I know this story before, so should I take a Nobel also?
Oh, you didn't know about genetic. If you knew about genetic and the story,
Yes.
maybe you connect this together and you got the point. So, you cannot see the DNA. Never ever, you cannot see the piece of DNA, this piece of DNA. But when you increase that one to billion pieces like this, just exponentially increase that one, after that you can see this part of DNA on a gel as a band. You remember when yesterday I told you this is a band from Y chromosome? You remember? It was PCR product. I used just PCR to check PGD. You remember?
Yeah.
And it is services. You get money. I said it is very expensive. You remember? It was just PCR. Is it understand? To see. That's the band. This is just only the band. I put the DNA here. DNA go to, because DNA has got negative charge, and go to the positive charge, and separated according to the size of DNA. And this is the DNA. And after that you can see that. You can see the DNA here. You remember it yesterday?
Yeah.
It was like that. You remember? And that's the DNA. What? Billion piece of DNA. Billions. Billion pieces of DNA. That's why you can see this one. Is it clear?
Yes.
Okay. So it is PCR. It is just only one method of 1,000 methods which we use in genetic. One of the application of PCR is coronavirus. You know that. Because coronavirus, you cannot see the virus, but you can increase part of the virus. For this application, to do amplification of the part of the virus. You cannot see the virus, but increase the amount of piece of the virus, piece of the DNA. Corona is RNA virus, but change to DNA and amplify that one. This is, that's RNA, RNA change to DNA, DNA amplify the piece of that. When you see the band, you say, okay, it is the virus. So you have the virus in your nose, for example. It's not just only for corona, it's for any kind of virus. You can make, using this one, you can make a lot of kits, detection kits. Exactly the same as they made for coronavirus, you can make for influenza virus and others. Is there any money for that? Cheat. You know how much money earned from detection of coronavirus? You can't imagine. You can't imagine how many money kits, detection kits, sold during that period of time, 2-3 years. Understand? This kit coming from science, technology, just a PCR. Just one technology, which is PCR. Understand? Based off that is just genetic. Is it clear? Do you remember I said about hosts?
Yes.
We have different hosts. For example, microorganisms such as bacteria, plant, or animal. I want to explain in this session about creation of genetic modification organism in microorganism, plant, and animal. Okay. What I said to you so far, I said about genetic engineering, manipulation of DNA. Am I right? I said just this one. What are the application of this one? The reason which you change the DNA. You change DNA for what? Which reason? It is this one. What do you do? What do you do when you become a medical doctor? Do you remember yesterday I said prevention, diagnosis, treatment. It is your job. Prevention, diagnosis, and treatment. It's job of medical doctor. That's it. I want to give you some examples from genetic engineering in prevention, diagnosis, and treatment. Is it okay with you? Okay. For prevention, the most famous product which you use for prevention is vaccine. You know vaccine. Definitely you got vaccine before. Which vaccine? Corona. Okay. Others. Polio. Mumps. Rabies. Tetanus. What? Pertussis. Different kind of vaccine. Polio. No? Polio. Many vaccines you got. Diphtheria. It's so many vaccines you got from, and all of them coming from genetic engineering. Understand? Is there money? Is there money in that? Yes. Definitely. Definitely. So, doesn't matter you are a medical doctor. Maybe you go for specialty of surgeon. Maybe you go for any sort of earning money. Depends. Depends on you. Which one you just make a decision. Okay, what you do here, what's the vaccine? Vaccine is a protein part of the body of the virus, part of the body of the bacteria. A protein. How we can make the protein? Do you remember? DNA, RNA protein. Exactly the same. I change the DNA, I put the DNA in bacteria. The bacteria make RNA. The bacteria make protein for me. Do you remember I said host organism? The bacteria is my factory to produce recombinant protein of the vaccine. Is it clear? The bacteria is my factory. The cheapest factory. I don't need to make a long plant and many things like these mobile phone factory. No, it's just a bacteria produce my protein, my favorite protein, which is protein of the vaccine, of the virus. Is it clear? Many different vaccine. You know this vaccine, for example, HPV. HPV, hepatitis B virus. You know HPV?
Yes. Papillomavirus.
Papilloma virus, human papilloma virus. Already we produce in Iran this one. My friends, they produce here in Iran. And it is produced in Gardasee, in Germany. And different products in the world. Is it how they produce? It is just a protein part of the HPV virus. A protein, but when produced in a bacteria, in a cell line. The cell line is my factory. Is it clear? Okay. Coronavirus, I said to you, the vaccine for coronavirus, different kind of vaccine they tried for coronavirus vaccine. Do you know what was the problem for coronavirus vaccine?
Mutations of the virus.
The strains of virus.
Why they spend a lot of time to produce corona vaccine? Why? Why they use RNA vaccine?
Because the virus mutated until its RNA, not DNA.
Because
Yeah.
virus was changing his genetic.
Because he changed his DNA always.
No. No.
Genetic reconfigurations. Just because.
No. Such as HIV. No. HIV is like that. No. The most important thing is that's coronavirus, for example. On coronavirus, we have proteins. You know protein? We have DNA, RNA, protein, and function, as I said to you. Okay, this protein has got amino acid, plus amino acid, plus amino acid. Is it right? Amino acid, amino acid, amino acid.
Yes.
It is not only amino acid in protein. We have something called post-translational modification. Do you know that? Post-translational modification. Do you know? What is post-translational modification? Is configuration of the protein. SS bound. And another one is, which is was very important for this virus, was glycosylation. Glycosylation. What this means? Means amino acid on protein become glycosylate. Glucose, fructose, maltose, and so on. And stick to the amino acid. For example, this is amino acid. And for example, this one, that's glycosylation on amino acids. And glycosylation of amino acid on this one changing a lot. and producing vaccine, antibody against that one is very difficult. Understand? I explained to you very easy, but in practice is very difficult. Understand? Explain is very easy, but in practice is very difficult. Okay. I said prevention. Now I am going to speak about diagnosis. Which kind of diagnosis? Kits. Do you remember I said about kit of diagnosis of corona? Kit of diagnosis of many things. Many. Have you done any checkup, lab test, blood lab test? Have you done it yourself, for yourself?
Yes.
All of you. Okay. Have you seen the report of the lab test?
Yes.
Give me one of that. One answer. About what? SGOT. AST.
DST.
Anti-DSD.
What?
Anti-DSD.
Antiviral. Any kind of that. All of the kits is designed by genetic engineering. How many kits do you use? Many. You are going to check, for example, hemoglobin A1C. Is it right? Hemoglobin A1C for blood sugar. Or enzyme of AST. Enzyme of PT. Any kind of enzyme, any kind of protein. Whatever you use is recombinant DNA molecule and recombinant protein in all kits for diagnostic lab tests. Many. That's the lab test. For example, alkaline phosphatase. Do you know alkaline phosphatase in your lab test? It's a protein. It's enzyme. Phosphatase means enzyme. How you can detect that one? With antibody, with antigen, and so on and so on. How you can make it? It is a protein. DNA, RNA protein. You understand what I mean? It's easy. So you can make protein, you can make recombinant protein, and you can make kits. Already many of these kits, I mean not already, before that, now it's better. We used to import from outside to Iran a lot, but nowadays we produce, many companies produce this. Another one is, another diagnostic kit on DNA is making probe. Fish, do you know fish? Yesterday I said to you, fluorescent in situ hybridization. We make probe. What is this probe? The chromosome is my chromosome, to be honest. They are my chromosome here. Because long time ago, 20 years ago, something like that, 2000, I suppose, 2004, 1999, 1998, I found a gene called TSJ10. This gene, TSJ10. I found this gene from testes, related to fertility and infertility. When I was in UCL, I compared normal testes with infertile testes, and I found this gene. And then I put the name of testis-specific gene A10 on that one. My supervisor told, put your name on that one, Hossein gene. I said, no, it is impossible. She died, but I said, I cannot put my Hossein name on this gene. Do you know why? Because I got this gene from testis. Hossein is a Hossein name. I said I cannot put Hossein name on that one. TSJ is better. So I put this name on this gene at that time. But I didn't know where this gene is on chromosomes. Look, I didn't know where is the gene on chromosomes. I made probe. Look at this. This one is probe. Look. Can you see? Yes. You can see here. Can you see here? two probes. The green one. They are probed. I made probe myself and put on the chromosome. And now you can see where is the gene is. Look, if this one is not here, is here, what happened? What this means? These two are here. This means we have translocation. Do you know translocation? If this one is here, means there is translocation. This is the normal one. We have, that's one allele, another allele. Another two, we have two chromosome 2. Chromosome 2. So we have two alleles. One allele on chromosome 2 here and one here. If these probes stick to here, for example, it means we have translocation. Understand? I did this one. And I used to, it's very nice to say. At that time, I used to produce the probe not only for this gene, for many other genes, many chromosomes, for Y chromosome to detect male and female. Because with this one we can say it is male or female with for Y chromosome. I used to make this probe for Y chromosome. This is Y chromosome. The price of the production of the probe for me was about just roughly about $5. $5 at that time was $5 to produce these probes for 10 reactions. I used to sell the probe to Wysis company, a company, very big company. To them, about let me remember, about $30 to $40. Look, for us, it was something about $5. We used to sell about $40. Do you know how much they sell?
100.
100?
400.
Something between. About $300. Look at that. This is the amount of the money I spent to produce that one. They sell this price. How much money between these two? Do you know what is this money for? For knowledge. Understand?
But they made so much more than you, and you are the one.
Of course.
They didn't do anything.
Yes. Because we were in the university. Yes, you are right. Because we were in the university, so I didn't care about the money at that time. I used to earn money, but the money become in my department, not in my pocket. If it was in my pocket, I said, no, I don't sell to you 400, I sell you 100 and better. But it was for university, I didn't care. Don't worry. Do you understand what I mean? But imagine how much is the price between these two, 5 to, imagine 3 to 300. It's the knowledge. It's the money for knowledge. And understand? It's that one. Okay. Another probe, it's for, I found another gene at that time, it was gamma 2. Do you know G proteins?
Yes.
G protein. I found gamma 2 G protein subunit. And at that time, I used to produce a probe for that one and to see where is the location of the gene on chromosomes. Anyway, do you know which chromosomes are? From mouse. Chromosome from mouse is like that one. It's very difficult to know which chromosome is chromosome 5, 6, 7. It's very difficult to find. Comparing to human. Human is easy. That's human chromosome. But this one is mouse chromosome. It's very difficult to distinguish between chromosome 3, 4, 5, 6 because all of them are the same. You can make probe for centromere. You can make probe for, do you know where is this part of the chromosomes? What's the name of it? Telomere. Telomere. Telomere probes, centromere probes, and many different probes you can make and sell it. What's the difference between fish and karyotyping? What's the difference between fish, as I showed you, and this is the karyotyping method? What's the difference?
That's what by fluorescence.
Sorry?
By fluorescence, it's show you the chromosome by fluorescence.
Yes, that one is what fluorescent, this one is not fluorescent. Do you remember yesterday my lecture about methods, about tests? This is the question. I said, it's your exam question. I'm telling you. What's the difference between karyotyping and fish? What's the difference? Fish, most of them check chromosomes. Most of them. But this one, check these bands. Each band is about 4 to 8 megabase, million base. DNA, do you remember DNA? 3.3 to 10.9? It's megabase, 8 megabase, each band. What about this one? This one. It is about 100 base pair, 100,000, 100 kilobase. This is megabase, this is kilobase, and this one is megabase. With karyotype, you can check megabase bands. With fish, you can check kilobase band. Which one is more sensitive? Do you remember sensitivity and specificity? Yesterday I explained to you. Which one is more sensitive? Which
Karyotyping.
It has higher sensitivity. Which one?
Karyotyping.
Karyotyping is more sensitive, or fish is more sensitive? Which one show you a smaller band? Fish. Of course, fish. It's fish. So which one is more sensitive? Fish. Of course. Understand?
So can you write the number of them on the
Which?
like
This megabase, can you see here? But for fish is kilobase. You can detect kilobases. 1,000 kilobase. In this one, you can detect megabase. Which one is more sensitive to check bases? Of course, fish, because you can detect a smaller size. Kilobase.
yeah.
With this one, you detect mega base. Understand? Okay. We are speaking about, we said something about prevention, detection or diagnosis. Now I am saying about treatment. One of the treatment is drug, pharmacogenetics. Do you remember yesterday I said a lot about pharmacogenomics, pharmacogenetics and so on. Do you remember I said about drug design drug. There are personalized medicine and personalized drug. Yesterday I said a lot. So for treatment in future we can use DNA for design drug. That's the production of the recombinant. How we produce recombinant? 20 or something years ago, no, 18 years ago, something like that. 18 years ago when I back to Iran, at that time, I decided to make a factory. That's my factory. The picture of the factory which I made. I start to make a factory for production, recombinant protein production. Recombinant protein production, such as TPA, insulin, HGG, interferons, and many different. Not I produce all of these. No, just one of that is enough. So, when you are speaking about production of the recombinant protein in a host, you need a lot of things. You need fermenters to produce host in this one. We produce bacteria here. It is centrifuge because you have to separate proteins after that and purify the protein. And then many other things you have to produce, and you have. This factory should be in clean room. Anyway, I suggest to you go and see many factories. It would be useful for you if you want to go for this way. Maybe, I don't know, maybe you go become a surgeon, you become an internist and some other thing, but maybe you go like me to this way. After my medicine, I started neurosurgery first. After finish my MD in Tehran University, long time ago, many years ago, I started neurosurgery. A little patient. Most of my patients die due to tumor in brain tumor or accident, car accident, tumor, motor accident, and so on and so on. I said, I don't like this specialty. I changed it to some genetic exactly. I changed it 2 weeks or 3 weeks I go for neurosurgery. And then no, it's not my favorite. I don't like to see dead person. No, I don't like this. I changed it. I said I like money. So I changed it to I am honest. I changed to this one. At that time they told me, oh, you did wrong. I said, no. It is, I suppose it is my favorite. I don't like that one. Okay, that's the factory. You can produce many drugs. Look at this. Drug for cancer, drug for vaccine, recombinant protein for many things. You can produce recombinant protein in these sort of factors. Look at this one. It's easy, I mean synthesized but easy for genetically engineered organism production. One of the most famous, to be honest, most famous drug recently, most famous drug recently is monoclonal antibody. In future, when you finish, definitely you will be involved in treatment with monoclonal antibody. When I finished at that time, long time ago, when I finished my medicine, there was nothing called monoclonal antibody. At that time, we didn't have monoclonal antibody. But now, we have a lot of different kind of monoclonal antibody. Do you remember yesterday I said to you, a treatment of breast cancer, Herceptin, trastuzumab, stick to receptor, and it is monoclonal antibody. Do you remember? It's one of that. But many monoclonal antibody produce. What is monoclonal antibody? Monoclonal antibody is, you know antibody, definitely you know antibody because you passed immunology. So, this is antibody. It is a big protein. It is useless for treatment. What is monoclonal antibody? Monoclonal antibody is just this part of the antibody. Look at this one. Just this, a small part of the antibody. This part. The part of the antibody can stick to the antigen. It's just exactly very little part. And it is just very small amino acid. 10 amino acid. 8, 10, I would say 20 less amino acid. You see, it is monoclonal antibody. Recently, they use a lot of monoclonal antibody. A lot. 400 MAB. MAB is monoclonal antibody. Is brief of monoclonal antibody. We have a lot of drugs called MAB. Do you know? Yesterday I said trastuzumab, Herceptin. That's monoclonal antibody. That's many MAB, down there. More than 400. 100, absolutely more than 100. I have to say 200 of that approved by FDA in the USA. It means we use also in Iran and we produce some of that. Many monoclonal antibody are already used for different disorders, not only for cancer, even for knees. No, for knees. For knees. The pain in knees for after cartilage degeneration for old people, they use for rheumatoid arthritis. For many disorders, many disorders already we use MABs, different kind of MABs. When you finish your MD, I am sure it become not 400, this is 1,000 of monoclonal antibody. All of them definitely produce using genetic engineering. Understand? Because you are going to produce just this part of the antibody, not all of that. You put this part in DNA and transfer to the cell line and produce antibody. Just to show you for one drug, such as this one, Humira, this one, adalimumab. Adalimumab, it's one of the drugs. In 2015, not now, 2015, is top 10 monoclonal antibody by Cell in 2018. It is this one. How much?
20,000.
20,000 million dollars. $20 billion. How much oil we sell? How much oil we sell in this country? Comparing to this one, like that. That's it. You want this one to see this one? 2019. That's the cell. This one is 2024 now. 2025, 2024. This is the cell of monoclonal antibody. Annual cell of million. How much is 2024? 200 billion dollars. $200 billion. Just monoclonal antibody. Just monoclonal antibody. I'm not saying about other drugs. I'm not saying about recombinant drugs. I'm saying just about monoclonal antibody. How can you do? It's a way we can produce. Instead of oil, we can sell this one. You understand what I mean?
Yes.
What is coming from? Come from under the ground? No, it's coming from here. It's coming from here. Inside. You know? The cells inside. It's the knowledge. You remember I said research, science, technology, production, and so. It's one of the production. Which way you make a decision to go? I don't know. You can go to be a good surgeon, neurosurgeon. It's nice, neurosurgeon. Anyway, I am not a neurosurgeon. I am just a geneticist. That's it. So understand? Can you see this one? To be honest, what I can say about my country, in my language is hot research. I don't know what you say in your language. It's absolutely disappointing. Why? Because we can earn money from knowledge. Okay, let's go to another one. Another treatment. Do you know this one? Stem cells.
Yes.
Do you know stem cells?
Yes.
Stem cell is sort of genetic engineering also. Stem cell. It's a stem cell. This one is a stem cell. That one is a lot of a stem cell. A stem cell. How many kinds of stem cells do we have? How many kinds of stem cells do we have? A stem cell is the cell can change to make many different cells. You know that. It's undifferentiated cell. Undifferentiated cell. Okay. How many kinds of stem cells do we have? You know?
Embryonic stem cells.
Exactly, you are right. Embryonic stem cell.
And adult stem cells.
Adult stem cell. Correct. And one more. No. One more. One more.
Pluripotent.
Yes. What?
Pluripotent.
Induced pluripotent IPS. Induced pluripotent stem cell. It means with manipulation, putting some genes inside the somatic cell, change the cell to stem cells. So we have three different stem cells. Embryonic, which coming from embryo, inner cell mass. From inner cell mass of embryo, inner cell mass. And adult, do we have adult? Yes, in our body is a lot of adult stem cells. Where is that, for example? Bone marrow. bone marrow. You can take bone marrow and use it. So, another one is IPS, induced pluripotent stem cell. Changing, manipulating, putting some genes inside the stem cell and inside the somatic cell and change the somatic cell to a stem cell. Is it clear? Okay. So we have different. And the knowledge for stem cell is also genetic engineering, based on genetic engineering. And we can produce skin, for example, from a stem cell. From stem cell. Regenerative skin, regenerative tissue, a lot. Another application, what I am saying right now, I am explaining about, do you remember, application of genetic engineering? I am saying now about, I said some application in prevention, I said some application in diagnosis, now I am saying application in treatment. One of the application in treatment is gene therapy. Next session I will explain. Okay. Another application of genetic engineering. Do you know, this is a host. Any bacteria, yeast, cell line, or animal, or plant. What we can do with this host, it has nuclear genome. Do you remember yesterday I said nuclear genome? Is it possible to delete one gene from this? Yes. Or is it possible to add one gene to this one, to genome of this host? Yes. So we can add one gene to this host, to genome of this host, or we can delete from this one. What's the name of the adding a gene to this one?
Transgene.
Transgene, exactly, transgene. What's the name of deleting of that? Knockout gene. Knockout host. Knock out, take out. Another one is knock in or transgene. Is it clear? Which gene is there? Do you know GFP, green fluorescent protein? What is green fluorescent protein? It's a protein. What is coming from?
Fireflies?
Starfish. You know starfish in ocean? Starfish. Because they produce fluorescent under UV. Fluorescent. This protein under UV become fluorescent. That's from starfish, something like that. Green fluorescent protein. It's a gene. It's a protein. If we put this gene in the genome of the host, what happened? Become inside the genome, and it is a transgene. Produce RNA, produce protein, and the function of protein is green under UV. Is it clear? Or not? Produce the protein in all cells of this host. Monkey host, mouse host, cat host, any kind of protein. They can produce GFP. It is just for experiment. It's not for production. Is it possible to put a gene in a host that produce a protein in the milk of the host? The milk of the host. Is it possible? Yes. This one is produced in all cells. That one can produce in the milk. That one produce in eye. Can you see? Green fluorescent protein in eyes. Is it possible to produce protein one protein in the milk. What means in breast of the, for example, cow, breast of the sheep. Is it possible?
Yes.
Is it possible to put what, do you know growth hormone?
Yes.
Growth hormone, you know growth hormone. Is it possible, for example, the fish has got growth hormone. Is it possible we put, instead of one growth hormone gene, we put two growth hormone gene?
Yes. Possible.
Produce more growth hormone in the fish. What happened?
More big.
Instead of this size, fish is this. Is there any money inside?
Yes.
Good money. A lot of money. Yes, you can see that one. Here. You didn't change anything. It's not growth hormone from other species. It's growth hormone from exactly the same fish. Exactly human. We have one human like me, small one. Somebody else has got more gene this size. What's that? Bodybuilding one. Yes, more hormone of something, androgen and something, any kind. Understand? Is it possible to do that? Yes. Is genetic engineering. What I'm saying about genetic engineering. Is it possible to produce, for example, in cow, in sheep, or in goat milk, produce a protein in goat milk? Yes, it is. That's farming. Do you know farming? Farming with F is produced in farm. But this is farming from pH. Farming means production of pharmacology, drug inside the, such as animal bioreactor. I said in genetic we use animal as a factory. Here is animal bioreactor or farming. Understanding this terminology? You must learn about this terminology. I said factory, it is saying animal bioreactor or farming. This is our factory. Understand? Producing in transgenic goat human antithrombin or do you know thrombin? It's for clotting. Anti-clotting. Produce. What's that useful for?
People with viscous blood.
For MI. MI. You know MI?
Yes.
MI. And they inject to destroy clot. Am I right? Okay. It's very expensive. Produced in goat milk. But you cannot kill this goat and make kebab and eat it. Because the price of this goat is $1 million. Understand? You have to grow up this one and have a lot of babies and so on and so on. And when they are 100 or 1,000, you can extract the protein from milks, tons of milk, not just a small amount of milk. For example, you cannot produce this one in mouse milk because mouse milk is very small. Milk in mouse you can generate a lot of mouse quickly, but the milk is I don't know. I suppose it's very small mouse milk. You can not do that. So you need this one. However, period of the life, lifespan of this one is much higher, longer than mouse. what we have to wait? Produce spider silk. Do you know spider silk?
Yes.
Spider, Spider-Man, spider silk in goat milk. What's that for?
Is it because spider silk is very strong?
Okay.
So it can be used for maybe construction.
For they use for
Clothes? Silk?
To repair wounds?
bulletproof gloves. You know bulletproof
Yes.
or bulletproof? Yes, they use for that. So it's very, very expensive. Very expensive. So spiders here. But it doesn't mean you kill this goat and eat it and you become Spider-Man. Don't speak funny. Then kill this one and make kebab and eat it and okay, change to Spider-Man. No. No. Somebody told him here, one stupid man told, don't eat this one. You become a Spider-Man. Oh, you are stupid. The price of this goat is $1 million. Kill this one and eat. Anyway, then said, if you eat this one, you become Spider-Man. Watching TV a lot, I suppose. Anyway, producing a lot of proteins, insulin, hemoglobin inside the animals. That's the application of genetic engineering. It was transgenic. Let's explain knockout. If you knock out FOXN gene, what happened? What happened? If you knock out FOXN1 gene, what happened? The mouse is this one. The chachal mouse.
Like lose hair here?
Sorry?
Lose their hair?
Doesn't matter, lose hair. Okay, yes, lose hair. What doesn't matter?
Going green, I think.
No, it doesn't have thymus. What is that for? It is very good mouse for because doesn't have
sensitive
immunity.
Yeah, it doesn't have
This one doesn't because doesn't have
time.
thymus, so it doesn't have immunity. It is useful for transplant any kind of tissue, such as tumor tissue. You can transfer human tumor tissue to this one and check what? Check efficiency of chemotherapy. Efficiency of chemotherapy. You cannot check efficiency of chemotherapy on human. Never, ever. You can check efficiency of chemotherapy on this one. Because this mouse doesn't reject human tissue, such as this one. It's real. It's not a picture. It's not by AI. It is real one. Produce human cartilage, human tumor. Understand? Is it useful? It is knockout gene, knockout mice. Already we have in Iran, and maybe when you go to Tehran, it's in Imam Khomeini Hospital. We produce this one. If you want to do something with that mouse. Another mouse is a skid mouse. Severe combined immune deficient mouse. Doesn't have immunity. So it is also useful for tumor implant tumor tissue and check the efficiency or effect of drugs on that. Understand? Is it clear? Okay. Again, that was about animal host. Now I am saying about plant host.
Can we take a break?
Sorry?
Break? 15 minutes break?
Yeah, sure. Okay.